just kind of chose some really big ones that regulate these guys. So the final exam itself, so it is on Monday for you guys. It's going to be about 60% new material, 20% exam 1 material, 20% exam 2 material. So 40% old material, 60% new material. What else we got? Any questions? The final is, I didn't post that for you guys, did I? I did it for my back in one. So yeah, I'll post where it is. I don't think it's in here. I want to say it's in a different room, but I could be wrong. I could have been looking at a different proctoring exam. So I'll double check that, and I'll actually send out an announcement about that. I think it's at 12:30, though, on Monday. Wait, no. I need to check. It's a week away. Well, not even. Okay. Good question. I will send out an announcement about that. I do, but I don't remember. It is at 12:30. I got it right. Okay, yeah, 12:30 on Monday. Are there any burning questions? Do you guys want me to go over kind of the transcription, translation, and then the regulation stuff, just to kind of set that in your mind a little bit more, or is there something else you want to do today? What is it? Yeah, okay. So I will, maybe we can do new material since it's kind of hopefully in your head today. We can focus on older material if you want tomorrow, or if you have any burning questions, we can do that. So I'm just grabbing my pen, actually. Here's one. All right. So I'm just going to pull up like a whiteboard and just kind of write things out as I go. Is this right? Is this work? Oh, no, it doesn't work. Sorry, I do have a pen. Like I said, just one of those days. Getting pulled into stuff I don't want to get pulled into. All right. Starting with, all right, so we talked about DNA replication. We're not going to talk about DNA replication right now. We're going to talk about DNA, starting with DNA, because we got to make the message RNA. So first thing, I want to make sure you guys understand the difference between transcription and translation. So transcription is DNA to mRNA, as we know. And if this feels like you know this, then sit back and relax. So we got transcription going on. Where does transcription take place? Nucleus. So with transcription, there are different ways you can regulate transcription. So with transcription, you have, so this is, let's say this is a gene that we want to turn on. Let's say we can do the insulin gene. Remember, there's a start site. This is going to be +1. This is going to be the start site of that gene. And then remember that there is a promoter region that's upstream from the gene. This promoter region, and this is going to be important for the regulation, because the promoter region has what's called a ta-ta box. Ta-ta. And that's called ta-ta because it's got a string of T's and A's in that section. But this ta-ta box is important because this is going to be where your transcription factors bind. So TF2. TF. Whenever you see this in science, TF usually means transcription factor. So transcription factor. Transcription factors help with transcription. And TF2, this 2 part, stands for RNA polymerase 2. Because remember, you have different RNA polymerases. But the one that's going to make the mRNA is RNA polymerase 2. RNA polymerase 1 helps make the ribosomal RNA, and RNA polymerase 3 helps make the transfer or transport RNA. But this is the big one that's going to make the messenger RNA. And in order to get RNA polymerase recruited, you need transcription factors. So this is a regulation here. If these transcription factors, TF2s, don't bind here at this promoter, more specifically the TATA box, RNA polymerase won't come. And so that can regulate what genes are going to be expressed. Only the genes that have the right transcription factors will be expressed. And so this promoter region, this is going to be important pre-transcriptional regulation. So again, I'm kind of combining the last 3 lectures. So the first lecture was on transcription, the second lecture translation, and that last lecture was on regulation. So the last 3 lectures, I'm kind of just giving an overview of all 3 of them. So this is pre-transcriptional regulation. Again, it's regulated in that if these transcription factors don't bind properly, then RNA polymerase will not be recruited. You gotta put these guys on the TATA box, and then RNA polymerase will bind, and then it can do transcription.
Can you repeat that last little chunk?
I can try to remember what I said, but essentially, the transcription factors have to bind to this promoter region, and within that promoter region is this TATA box, and that's technically where the transcription factors bind, and these are required in order to recruit RNA polymerase 2. So without these guys, you don't get RNA polymerase and you won't get transcription. So that's just a pre-transcriptional regulation. And the reason I say pre means before, because we have post-transcriptional regulation we're going to talk about as well. That's going to be the splicing that we talked about. But this is just pre-transcriptional, so now this is required for initiation of transcription. Elongation of transcription is simply just making the mRNA. There's nothing going on more than that. So step 1, we make the RNA. Step 1 is we put the transcription factors on here. Now RNA polymerase is going to make the messenger RNA. And so now we have this mRNA. But this mRNA transcript is not done yet. We're going to have to edit this mRNA. This is just an mRNA transcript. I don't know why it keeps darkening like that sometimes. So remember, we have to do a couple of things, and this is going to be post-transcriptional regulation. So post-transcriptional regulation is what we're going to talk about next. I'm going to back this up for a second here, though. And so initiation of transcription is the binding of the transcription factors. RNA polymerase binds, that begins transcription. Elongation is just RNA polymerase to making the mRNA transcript. And termination of transcription, there's lots of ways eukaryotic cells will terminate transcription. The termination that I told you guys to remember, the most common one is called row-independent termination. And that row-independent termination is where you have a long strand of G's and C's, G, C, long strand, and they're going to create a hairpin loop, and that's going to cause RNA polymerase to fall off. So that's how it's actually terminated. And that's just one example of how eukaryotic cells will terminate transcription. There's a whole host of other ways. But this is a really common one. It's called row-independent termination. And that's creating a hairpin loop because there's a long strand of G's and C's that will attract each other and come together to make that hairpin. Right. So again, that was transcription. Now let's focus on after transcription has happened. So after transcription, we have this mRNA, and sometimes we call it pre-mRNA because it's not been edited yet. So we need to edit this guy. First thing, and this actually happened earlier, but I'll just do it here. We have to add something to the start of this pre-mRNA, and that's called a 5' cap. It's technically called a 7-methylguanosine cap. But for short, we just call it a 5' cap, and that's because it is a 5'. So this is the 5' end of that messenger RNA. And then we also have this poly-A tail. Poly-A tail. And this poly A tail is also going to be part of that editing. So both the 5' cap are important for regulation, and poly A tail are important for regulation. Without the 5' cap and the poly A tail, this would be degraded rapidly. And so this keeps that gene, that mRNA sequence around longer, making it more likely to be translated. So if without the 5' cap or the poly A tail, this guy would be degraded. It would likely not even make it to being a protein. So that's why this is important for regulation. And this is post-transcriptional regulation because it's happening after transcription. That's what the post means. And so 5' cap and poly A tail, and the other one is splicing. That's another post-transitional regulation. Because remember, we have to splice this guy. It's got introns and exons. Introns and exons. Which one of these needs to be cut out? Introns, exactly. The introns have to be cut out. And the exons glued back together. So that's what splicing means. Introns are cut out and exons are kept. Introns cut out. And the exons are kept. And this EX stands for expressed. So these guys are going to be annealed back together. And that's what the splicing is via spliceosomes. And that's really transcription. At this point, we should be ready for the next part, which is going to be translation. So that's kind of like a snapshot of that first lecture, along with the last lecture with the different different regulations. So we have two examples of regulation here. One example was pre-transcriptional regulation, and that's the whole top box and transcription factors. The other one was post-transcriptional regulation. That was the 5' cap, poly A tail, and splicing, are just three examples of post-transcriptional regulation. All right, any questions about transcription, how it starts, how it stops, how the pre-mRNA transcript is edited? Yeah.
So TF2 binds to the top box, and that allows for RNA polymerase 2 to bind there and start transcription.
Exactly.
Okay, and then the 5' cap and the poly A tail, those are added on after it's been transcribed from DNA.
Yes.
And this all happens in the nucleus, that too.
Yes.
Okay. We just need to know that the 5' cap and the poly A tail are just added on afterwards to allow it to sit.
Maintain stability.
Okay.
Yep.
And then it's also getting spliced too. So it's getting all three of those.
Yeah, and the 5' cap, technically speaking, is added on almost immediately onto the mRNA as it's being transcribed. It's still post-transcription in that you have to make the mRNA first, and then the 5' cap is put on. And then once the mRNA is made, the poly A tail is added. So technically, it's not happening all at the same time, but it is happening post-transcriptionally in the nucleus.
Do we need to know like what's doing the splicing? Like what's coming it out?
Yes, what is doing the splicing? What's that thing called? Spliceosome. It's called a spliceosome, yeah. But yeah, that's basically it in a nutshell. All right, so the next part of this, now that we have our official mRNA, I don't know why I just put a P there. Let's try that again. Now that we have our mRNA, this guy is going to go out of the nucleus. So it's going to leave the nucleus and go into the cytosol. And then we're going to do translation. So translation. And translation is where we take the mRNA and we're going to convert this guy into a polypeptide. And the part that you don't need to know because you learned last term, but it's good that you should remember, this guy is going to be folded in order to make a protein. So the polypeptide is just the primary amino acid sequence. Amino acid sequence. And that primary amino acid sequence will be folded, so you have the secondary folding, tertiary folding, until you make that active protein. But we're going to focus on this part here, the mRNA being converted into the polypeptide. That's what translation is. Translation has nothing to do with the folding. All right, so this is going to require all of the RNAs. So it's going to require the tRNA, which is carrying what?
Proteins.
The amino acids.
Yeah.
And it's also going to require the ribosomal RNA, which is going to be important, a component of the ribosomes. So you need all your ribosomes for translation. You need your messenger RNA for translation, your tRNA, and your ribosomal RNA. Transcription only requires a messenger RNA. All right, so how it starts. So remember, we're in the cytosol. We could be in the endoplasmic reticulum. Remember, ribosomes are associated with the rough ER. That's why the rough ER is rough. It's got ribosomes. But really what we need are just ribosomes. We technically don't even need the endoplasmic reticulum, well, depending on the protein. Ribosomes, however, are required for protein synthesis. You cannot make proteins without it, and that's because they're going to facilitate all this. Ribosomes are going to read, no Siri, I don't care. All right, ribosomes are going to read the messenger RNA and match it with the correct tRNA. Correct. That's what ribosomes are going to do. They're going to read the messenger RNA and match it with the correct tRNA in order to grow that polypeptide chain. So with this, it all starts with the ribosomes. That 5' cap I talked about a minute ago that helps with stability, it also has a function of helping the ribosome recognize the messenger RNA. So that's just another function of the 5' cap, helping the ribosome recognize the mRNA. And essentially, the ribosome actually is different sizes. We're just going to do the large and small subunit, and I don't care if you remember the sizes. So both of these here are the ribosomes. And there's rRNA associated with both these big and small ribosomes. So again, this whole thing, this whole blue, I'm going to make it red. The red is the ribosome. And the messenger RNA is actually going to be inside this guy. So this thing is going to be the mRNA. The green thing is the mRNA. Now, what's going to happen, the mRNA, remember the mRNA has a codon, this 3-letter codon. I'm going to blow this up so I can do a 3-letter codon. Remember the start codon. What is the start codon for mRNA? AUG. So this mRNA actually has a codon that's, I shouldn't have made it red and blue. All right, let's check it out. AUG. This is the codon of the mRNA. And they're all in triplicates. So AUG is going to have to match the tRNA that's coming in. So I'm going to make this tRNA yellow, because I'm running out of colors. So this tRNA is yellow, and it's coming in, and it has on it the amino acid that it's carrying. So it's got its amino acid, and what is the amino acid that's going to be the first amino acid added? Methionine. Methionine. That's spelling. All right, so this AUG has to match it, and that's going to be, what is A going to match to?
T.
Not U. You're correct if we're talking about DNA, but we're talking about RNA. So U, A, C. So that's going to match the AUG, and the tRNA is actually called the anticodon. So the anticodon has to match the codon. So the tRNA has an anticodon, UAC, that matches the codon of the mRNA, which is AUG in this case. And that's the one that you need to remember. The start is methionine. So that's going to be the first amino acid that's going to be added to this polypeptide chain. And that's what the ribosome does. The ribosome is matching the mRNA sequence to the correct tRNA. And so the next tRNA that comes in, let's say it's serine. I actually don't know the code for serine, but let's say that's what it's carrying. Then serine is going to be added to the polypeptide chain, and then tyrosine, and then isoleucine. So essentially, we're going to be growing a polypeptide chain. That's what the ribosome is doing. It's matching the mRNA to the tRNA. And what is going to facilitate the initiation of this, the regulation? So for example, we do have a pre-translational regulation here. And the one we talked about, a student actually called it ELF2, which worked for me. But eukaryotic IF2. So it's technically not an L there. It's eukaryotic initiation factor 2. This is a pre-translational regulation. In other words, this ELF or EIF2 is going to help the ribosome match the mRNA to the first tRNA. And without EIF2, initiation of translation will not occur. So EIF2 is important for pre-translational regulation. This molecule, it has to be phosphorylated. Well, actually, when it's phosphorylated, it's inactive. So when it's dephosphorylated, it's active. I don't care if you remember that part. I just want you to remember that one of the pre-translational regulations, and there's many, this is just an example of a pre-translational regulation is EIF2. If this guy is not active, translation won't occur. Even if the mRNA is there and the tRNA are there and the ribosomes are there, if EIF2 is not active, translation won't occur. And that's a way your cell can regulate which transcripts are actually translated. All right, so that was the EIF2. That was a pre-translational regulation. It's the only example I gave you guys. So for transcription and translation, there's always initiation, elongation, and termination. Initiation is just this. EIF2 helps the helps the ribosome match the messenger RNA with the tRNA. That's initiation. Once initiation has happened and methionine is added, then we start growing the polypeptide chain. Essentially, this mRNA is going to read the mRNA transcript 3 nucleotides at a time. 3 nucleotides at a time, and it's going to start adding amino acids in order to grow a polypeptide chain. And that is the whole point of translation, is to make this polypeptide. And that's elongation. There's not really anything to elongation. But there is something to the termination, which we're about to get to after this question. Yeah. No. Ribosomes. That's a tRNA. It's the ribosome to read the mRNA and match it with the correct tRNA. So here's the, well, yeah, there's lots of tRNAs hanging out up here. And here's all these tRNAs, and the ribosome has to match it with the correct one. We could watch a video if I get the computer going. Watch Amoeba Sisters, guys. Amoeba Sisters, this will give you a good animation of this. this computer wasn't working when I first turned it on. But yeah, that'll help. Yeah.
Just a quick thing, you said ELF2 helps the ribosome match the right tRNA to the mRNA.
Yeah.
Okay.
Yep.
Then that's
And so now initiation can occur. But without ELF2, initiation can't occur because EIF2 has to be activated.
you said even if you're not
Yeah. Yes. Okay. All right. So yeah, there's actually a bunch of these tRNAs that are going to be hanging out, and each one of these tRNAs, guys, has like a serine or an isoleucine or whatever it is attached to it, alanine, and these amino acids are waiting to be grabbed onto by our ribosome. The ribosome is having to make sure that he's picking out the right tRNA to match it to the correct mRNA codon. We can make this more complicated. Yeah. Like I said, we can. We don't need to know that. But yeah, there's going to be a very precise method that this occurs. This is all like this is biochemistry. And we can, like, there's chemistry involved in all of this. There's moving of electrons, and there's energy, and there's ATP, and there's other factors, like protein factors, that facilitate this. I'm giving you one example, which is ELF2, but there's tons of others that are actually involved in this. And the Amoeba Sisters, the reason why I'm recommending it is because they won't go into a lot of detail, but they'll give you what you need to know, and you'll see it in animation form. They might not talk about ELF2, though. They won't go, but I don't think they talk about regulation. But you guys still need to understand this is regulated, so I have, like I said, picked out a few regulatory steps that you guys are learning, like ELF2. That's
when it starts, it's RNA that we're making, is it?
no, we're making a polypeptide, not RNA anymore.
Well, the polypeptide that we're starting to make, is it, are you using
Methionine is the first amino acid for that start codon AUG. Always methionine is always the start amino acid of that polypeptide. Okay, so we've done initiation and we've done elongation, and we have made, now we're getting to the end of this mRNA transcript. What is this? There's going to be a stop codon here. What is the stop codon you need to know? UGA. And when the stop codon makes it to the ribosomal RNA, this is going to terminate translation. So as soon as this UGA makes it to this ribosome, that will terminate translation, and we will officially have our polypeptide. And this polypeptide is going to have other things that's going to affect it before it's folded, but we're just going to talk about one post-translational regulation. Do you have a question? There's 3 of them that you can memorize. I just told you to memorize this one because this is where I went to undergrad. But you can learn the other ones too. The stop codon and one of the stop codons. So you need to know the AUG and the UGA. But they're technically 3 stop codons, correct? So it's not just UGA, but others can stop this as well. All right.
So the polypeptide is the final product.
Yes, the final product of translation is the polypeptide. And again, it always starts out with methionine, and then it's got a whole host. It could be serine, alanine, isoleucine, leucine, valine, blah, blah, blah. And so the polypeptide chain is just a long chain of amino acids, always starting with methionine. And it depends on the gene. If this were insulin, it's going to be a different polypeptide than glucagon. The The only other regulation, so this is the end of translation. The only other regulation that I have for you guys, well, actually, I didn't talk about epigenetics. I'll go back to that one in a second here. But the post-translational regulation I told you guys to remember is something you already learned. Translational. And that is ubiquination. Ubiquination. When you ubiquinate, when you ubiquinate, add this molecule to a protein, and I'm just going to add a UB. When you add ubiquitin to a protein, it tags it for degradation. So we do call it ubiquination. When you add a ubiquitin to a protein, you tag it for degradation. And that's a type of regulation. That is a post-translational regulation. Because essentially, if you get rid of a protein quickly, then it doesn't stick around in the cell very long, then it doesn't do a whole lot. But if that protein sticks around for hours, weeks, days, years, then that protein is going to be doing a whole lot. So one way to regulate expression of genes is by determining how long that protein is going to stay in the cell. If you tag it with a ubiquitin, then you tag it for destruction. And that means it won't stick around very long. So this has to do with protein stability. This has to do with protein stability. Just like the 5' cap and the poly A tail had to do with RNA stability, ubiquination has to do with protein stability.
Are you tagging it to be degraded because you just took it out, because it got pulled out?
You're tagging it for degradation because it just depends on the cell. If the cell only needs this protein for an hour, then it can tag it for degradation. But if it needs this protein for a whole day or a week, then it won't tag it. So it's basically about protein stability and how long that particular protein needs to stay in that cell. And that depends on the cell and what it does.
There's something else that goes with ubiquination, because when they tag it, there's something else that goes with it.
Well, so ubiquination has to, means it's going to be degraded by proteasomes. But lysosomes can also do protein degradation, and they don't have to be ubiquinated. Proteasomes, exactly. So, what did I forget here? Oh, the only thing I forgot with regulation, and I didn't forget it, but it has to do with epigenetics, and that was, there's 3 things with epigenetics, and this has to do with HDACs and HATs. So, what does the H stand for? Histones. And the A stands for acetylation. So the HDAC, D stands for deacetylation. So when you deacetylate something, you get rid of the acetyl group, versus HAT means you do acetyl something. You add an acetyl group. And when you add an acetyl group, that basically loosens the chromatin. So an acetyl group will loosen the chromatin. And when you loosen the chromatin, that means you're going to have gene expression, or the potential for gene expression. And this is a way your cells will regulate gene expression. Versus HDACs, when you deacetylate, when you take off the acetyl group, then this is essentially going to condense or tighten the chromatin. Condense means tighten or pack. And that means you prevent gene expression. And the other one was methylation. Another form of epigenetics is methylation. When you add a methyl group to the DNA, do you silence it or do you prepare it for expression? silence. You silence that gene. And these are all examples of epigenetics. And so that has nothing to do with the DNA itself. It has to do with whether or not the stuff, the important machinery can get to the DNA in order to express those genes. And in this case, they will not even be able to get to the DNA to express those genes if they're methylated or deacetylated. No. HDACs are histone deacetylases, not methylate. The A stands for acetylation. So epigenetics are all about acetyl groups or methyl groups. HDACs and HATs are acetyl groups. And methylation is just methylation. There's no acronym for that. Yeah.
What does methylate?
So methylation, when you methylate the DNA, you prevent it from being transcribed. So it silences the gene when you add a methyl group to the DNA. Huh?
We need to know what does that to the DNA.
Oh, yeah, no. I don't, there are enzymes that will do it, but no.
All right, cool.
Yeah.
Thank you.
Yeah, so HDACs versus HAT, you should know the full name. The H, you should know what the H stands for. It's histone. And A stands for acetyl group, and the D is histone deacetylase or histone acetylase. And the T stands for transferase, which is the enzyme. But I will spell I will write these out. I won't just put HDAC or HAT. I'll put histone deacetylase transferase on there. I'll write it out, the acronym. I don't expect you guys to know the acronym, so if I do put an acronym on the exam that you don't know, then you can ask me. I'll let you know what the acronym is. Unless it's, I don't know, like DNA. You should know what that is. There might be some acronyms like DNA, RNA. I can't think of anything else right now. But there's some acronyms you should know. But that one, I'll write it out. They do, they're both forms of epigenetics. So acetyl groups and methyl groups are forms of epigenetics in that, but they do different things. So a methyl group has nothing to do with histones. So when you methylate, you're actually methylating the DNA. And when you methylate the DNA, you prevent it from being expressed. Whereas acetyl groups, those are added to the histones, which is what the DNA is wound around. And so acetyl groups, that's why it's called the histone acetylases or deacetylases, because you're actually acetylating the histones, not the DNA. So the methyl groups, you're methylating the DNA. The acetyl groups, you're actually acetylating the histones. And that changes, they're both forms of epigenetics, but methyl groups and acetyl groups are two different things. Two ways to get to the same, I mean, you could say it that way, but it's both acetyl groups, so deacetylating and methylating DNA will both silence the gene. But I guess it depends on the cell's methods and why it's there, because remember, this can also be changed based on activity, whether you exercise, what your grandparents ate. But yeah, they will both silence the gene, just in different ways. Right, so that was transcription and translation, and all of these. So these are both, epigenetics is definitely, it's not considered pre-transcription, it's just considered a level of regulation that's above the DNA. Epi means above. So this is just another way the cell can regulate what genes are being expressed. It can do it from the epigenetic level, it can do it from the pre-transcriptional level, the post-transcriptional level, the pre-translational, and the post-translational level. And I gave you an example of each one of those, along with kind of a review of translation and transcription. That was kind of the last 3 lectures in a nutshell. And now, what?
10-minute break.
Oh,